Hello, and thank you for joining me for this webinar on codon optimization, how and why to design DNA for optimal soluble protein expression. By way of introduction, my name is Rachel Spear, and I'm a technical writer here at GenScript, focusing on our gene synthesis and molecular biology services. I did my PhD work in the neuroscience program at Weill Cornell, and my main research project there involved using a variety of recombinant fusion proteins for reported assays and biochemical analysis. So I would do the molecular cloning to create DNA constructs, then stably express them in mammalian cell lines, and then purify protein for IPs and Western blots. And my colleagues, who were doing in vitro enzyme kinetic studies, needed to purify large quantities of enzymatically active protein from an E. coli system. So as you might imagine, we ran into our fair share of concerns about getting the right DNA constructs and getting sufficient protein expression. And I'm a bit embarrassed to admit that at the time I was completely unfamiliar with codon optimization and gene synthesis, which may have expedited our experiments considerably. So I'm really excited that you all are attending this webinar today to learn more about codon optimization and how it can help your research. I see that most of our attendees are using E. coli expression systems, so I'll focus on E. coli, but I hope I've got something that will appeal to everyone. If you have a question at any time, please type it into the chat function. After the presentation, I'll read through the questions that have been submitted and do my best to answer them. This webinar, including the Q&A, will be archived on our website afterward. And at the end of the webinar, there'll also be a survey, and I'd ask you please to fill that out so that we can get some feedback on how useful you found this webinar. So let's get started. My goal today is to introduce you to codon optimization. First, some background on why codons matter for protein expression. Then I'll tell you about the codon optimization algorithm that GenScript has developed, which is called OptimumGene, and I'll show you some data on how it works and how well it works. And we'll look at some case studies from different research fields demonstrating how codon optimization provides a tremendous advantage for different types of experiments. And finally, I'll walk you through the solutions that GenScript offers to help you get better protein expression in your work. So to place codon optimization in context, here's an overview of a typical workflow for trying to express a protein. First, you need to select or design the amino acid sequence that you want to express, and then choose an expression host. Then design a DNA construct that will serve as your expression clone, then transfect it into your cells, express and purify your protein, and finally characterize it. Each of these steps is important in determining final protein yield. And at the end of this presentation, we'll return to this workflow so I can point you to some other resources for optimizing each of these steps. For today, we're going to focus on optimizing the codons within your expression clone. So why do codons matter? Well, as you know, there is redundancy in the genetic code, with 64 possible trinucleotide codons that correspond to only 20 amino acids, plus the stop codon. This means that you can introduce changes into the nucleotide sequence, most often in the third nucleotide of a codon, that will not change the amino acid. These are called synonymous mutations. And while synonymous mutations do not change the amino acid sequence, they do change protein expression levels. This fact has been observed many times in virtually every species. In fact, synonymous mutations can also alter protein conformation, post-translational modifications, protein stability, and consequently protein function, and thus the cell's behavior and survival. So when we design a DNA construct from which we will express a protein, we can choose to introduce synonymous mutations that will favor efficient, soluble, functional protein expression. That's our definition of codon optimization. So let's be very clear about what codon optimization is and what it isn't. In your expression clone, there are many different factors that you can choose to tinker with. As we've already discussed, you may want to change or shorten your amino acid when you select or design your amino acid sequence. That would be considered protein design, and it's very different from codon optimization because codon optimization only changes the nucleotide sequence but not the amino acid sequence. You may also want to try using different promoters or different affinity or solubilization tags. That would be considered part of your expression strategy, and again, that's outside the scope of codon optimization. What codon optimization addresses is the nucleotide sequence that will be translated into protein. So that can include your tags as well as your target protein. A lot of times people don't think too much about the tag sequences if they're already in the vector that you clone your target protein sequence into, but in fact we can and frequently do codon optimize these tags since their sequence will also influence your final protein levels. So the big question is, how do you choose the best codons to improve protein expression? What factors are at play here? Well, the first factor that people considered, starting decades ago, was codon bias. Now, there's a very rich and at times contentious literature on both the evolutionary history and practical applications of codon bias. So I'm going to keep this story simple, and if you want to dig deeper, I would refer you to the primary publications in the references slide at the end of this presentation. It has been observed that in every species examined to date, all codons are not represented equally throughout the coding regions of the genome. That is, there's a bias toward using certain codons and not others to encode any given amino acid. And there's a corresponding bias in the abundance of tRNA isoacceptors, so that the tRNAs for more commonly used codons are themselves more abundant. This bias is species-specific, so it's been noticed that when you try to express a protein from one species in a different host, for example, trying to express a human protein in E. coli, heterologous protein expression levels are often very low. And one explanation for this could be that when you have a mismatch in the codon bias of the gene versus the host, then the presence of codons that are rare in the host could reduce protein expression levels. And the mechanism could be that the relative rarity of those tRNA molecules could cause translation to stall. So maybe there's a long pause during the elongation phase while the ribosome looks for the right tRNA. So that's the theory of codon bias. Now, to try to address this problem, several different solutions have been used. The first is to co-express the genes along with genes that encode the rare tRNA molecules. So that essentially removes the tRNA bias in the host cell. This has worked pretty well in several cases. Another strategy is to alter the gene that you're putting in to replace the rare codons with preferred codons in the host. Now that can be done using site-directed mutagenesis, but as you can imagine, even with a relatively small gene, you might need to make dozens or hundreds of single nucleotide mutations, which could be very tedious and time-consuming. But it has been done, and it does seem to work. Later, however, as de novo gene synthesis has made it possible and increasingly affordable to simply synthesize from scratch any DNA sequence you can design, people haven't been constrained by a template DNA that they have to mutate. Instead, you can just do codon optimization to design your ideal sequence and synthesize it de novo using gene synthesis. So given that, codon optimization has become more popular more recently. So a 2008 paper, just to show you some data, used both of those two strategies to try to improve the expression of 30 different human genes in E. coli. They found that codon optimization increased the expression levels in 18 out of the 30 genes, and it also increased solubility in 8 genes. The study also used the strategy of expressing the rare codon tRNAs along with the non-optimized gene, and that increased expression of 15 out of the 30 genes, and 12 of those overlap with the 18 that were improved by codon optimization. So that suggests that codon optimization is working in part by overcoming tRNA rarity, but there are other mechanisms at play as well. And it's worth noting here that these researchers requested codon optimization from 3 different sources, of which GenScript was just one. And this paper doesn't present a head-to-head comparison of the different codon optimization algorithms. And also keep in mind that we've learned a lot about codon optimization in the 6 years since this study was published. So I think we would get better than 18 out of 30 now. So I think the biggest surprise has been that codon bias is not the only factor, and in fact, many cases, it's not even the most important factor for determining protein yield. So there was a very nice study published in Science in 2009 that investigated the relative importance of different features of the coding sequence in a fairly systematic way. And they did that by creating a synthetic library of 154 genes that contain different combinations of random synonymous mutations. But they all encode the same protein, GFP. So using green fluorescent protein allowed them to use fluorescence as an easy readout for functional soluble protein levels. Among the 154 constructs, they found 250-fold variation in GFP levels in fluorescence. But surprisingly, they found that codon adaptation index was not correlated with fluorescence. That is, the genes that were better adapted to E. coli's codon bias did not tend to have higher expression levels. But then they looked at many other sequence features, including the likelihood that mRNA will form secondary structures, such as hairpins, that might interfere with translation. And indeed, they found that the predicted mRNA folding energy in the 5' region of the gene explained 44% of the total variation in fluorescence levels across their library. That's nearly tenfold more than any other single predictor variable out of the dozen or more that they tested. So this demonstrates that rare codons aren't nearly as big a problem as hairpins in the 5' region of your gene around the ribosome binding site. So that it may mean that the hairpins are preventing the mRNA from being loaded onto the ribosome to initiate translation. Another study that changed our understanding of how to design genes for optimal protein expression is a Nature paper from 2012 that looked at the mechanisms of translational pausing. So they did a genome-wide analysis of translational pausing in bacteria by doing deep sequencing of mRNA fragments bound to ribosomes. Unexpectedly, they found that codons decoded by rare transfer RNAs do not lead to slow translation under nutrient-rich conditions. Instead, they identified sequences that are similar to Shine-Dalgarno sequences. And when those SD-like sequences were found within coding sequences, they caused pervasive translational pausing. They used a nice technique, an orthogonal ribosome that had altered anti-SD sequences, and showed that pausing is due to hybridization between the 16S ribosomal RNA and the SD-like sequences in the mRNA. So in protein coding sequences, they found, like throughout the E. coli genome, they found that internal SD sequences are disfavored, which actually leads to biased codon usage. So that they think that that's why among bacterial genes you see an avoidance of rare codons and codon pairs that because those happen to resemble canonical SD sites. And so the conclusion for our purposes of this paper is that internal SD-like sequences are a major determinant of translation rates, and so avoiding them can help reduce that ribosomal pausing and increase translation rate. So there have been many other studies that have identified other sequence features that affect protein expression through diverse mechanisms at different stages of the process, from transcription to mRNA processing and stability to translation initiation and elongation and protein folding. Now, in many cases, the practical implication of these studies is simply that we need to design DNA sequences to not contain certain sequences that have been revealed to cause problems, such as the SD-like sequences we just discussed, or AT-rich sequences that can cause premature transcriptional termination, or there are specific 7-nucleotide sequences that are known to introduce frame shift mutations, etc. Now, GenScript has an OptimumGene Codon optimization algorithm that considers dozens of different parameters that have been shown to influence the efficiency of transcription, translation, and protein folding. Our algorithm also allows flexibility to adjust the weight of different factors or to add customized constraints, such as filtering out restriction sites that you're planning to use in your cloning strategy, or reducing similarity between different sequences if you're creating a library. In some cases, you may want to specify an alternative codon table if you think your experiment can benefit from that. For example, there have been publications reporting that for certain amino acids, researchers have empirically observed a preferred codon that is not the most abundant codon. and that can be influenced by experimental conditions such as amino acid availability in your media. So some of our customers who order gene synthesis regularly will work with us to develop a standing protocol for codon optimization according to their particular needs. So we can definitely accommodate any special requirements that you have. GenScript has used the OptimumGene algorithm to optimize over 50,000 sequences in all major expression systems, which we're pretty sure is more than any other entity. GenScript was actually the first commercial supplier of gene synthesis services, and we've been in business for over 13 years, and we're still the largest supplier of gene synthesis in the world today. So that's how we've accumulated all this experience. And I think that our growth has been thanks in part to our continuous reinvestment in key technologies like codon optimization. So, how do we actually perform the optimization? Well, in fact, we hold a patent on our method for using a particle swarm optimization algorithm to power the OptimumGene software. And essentially what we do is an in silico evaluation of over 10,000 candidate sequences in each round of analysis, and we have a complex sorting matrix that then selects the best sequence for each gene optimization task. Of course, we're not the only company to offer codon optimization, so we routinely take our optimized sequences from the computer and into the wet lab, and we compare them to our competitors. And our in-house data suggests to us that the OptimumGene algorithm is actually more effective at increasing protein yields than our competitors' methods are. So here you can see two eukaryotic proteins that we're expressing in E. coli. For both of these, the wild-type sequence produced undetectable protein levels, so they're not even shown on this gel. When we ordered a codon-optimized sequence from a competitor, we could see pretty good expression, but our OptimumGene algorithm provided sequences that performed considerably better, so 3-fold better in one case and 13-fold better in another case. Now, our algorithm isn't static. We routinely test and improve it, both because we want to provide the best results for our customers and also because we use our codon optimization algorithm in-house for our protein services. So this is an example that our R&D scientists shared with me where they looked at a human gene they wanted to express in E. coli. They saw no detectable protein when expressing the wild-type gene, which is not uncommon, and in fact that's a major reason why researchers come to us to order codon-optimized synthetic genes. In fact, many researchers don't even bother testing the wild-type gene first, they just start with a codon-optimized gene to save time. So anyway, you see here that with our default optimization parameters in lane 5 that's labeled as Opti0, there's a pretty good signal. But when our R&D scientists tinkered with the OptimumGene parameters to create 5 alternative optimized sequences, they found a good deal of variation in expression levels. All of these sequences are a huge improvement on wild type, but some are better than others. And this variation is hard to predict until you actually do the wet lab experiment. So this is why some customers like to order multiple codon-optimized sequences and test them side by side to see which works best in their lab. But in most cases, people don't bother ordering multiple genes that are codon-optimized in different ways. Just a single codon-optimized gene is generally sufficient. So hundreds of papers actually cite GenScript for codon-optimized gene synthesis, and these papers really span a variety of journals and research fields, as well as a variety of reasons for pursuing codon optimization. You can find examples by searching the citations database on our website, which contains over 10,000 papers that cite GenScript services and products, and the ones that cite codon optimization will be under Gene Services. You can also see some different examples in the Gene Synthesis Handbook that we put out about a year ago. It was updated this past April, and it's available for free download on our website. So because I just love showing off how our customers use our genes, we're going to look at a few case studies in a little more detail. So this 2013 Nature paper studied the Neurospora gene called frequency, abbreviated as FRQ, which is critical for circadian clock functions in the Neurospora mold. They noticed that the endogenous FRQ gene exhibits non-optimal codon usage across its entire open reading frame, which is a little strange. So when they optimized codon usage, they saw an increase in FRQ levels. but they also saw conformational changes in FRQ protein and altered FRQ phosphorylation profiles and protein stability, and a complete loss of circadian rhythms. So this is pretty compelling evidence that naturally occurring codon bias can be biologically important, and not just in the sense that important proteins have been selected for high expression to contain the preferred codons, but as we see here, the FRQ gene has probably undergone selection to contain rare codons that slow translation down to allow the protein to fold in a certain way that's critical for its function. So I present this kind of as a caveat emptor for codon optimization. Increasing the protein expression level does not necessarily increase the level of properly folded or properly functioning protein. But another reason I mention this paper is because in this case the researchers needed a few different constructs that were codon optimized only in specific regions, which is easy to specify when you request codon optimization from GenScript. Okay. Another case study is a Nature paper from this past April where they set out to identify better anti-cancer drugs. And I really like this paper because they combine chemical proteomics, pharmacology, and genetic approaches to really go full circle from a drug screen to a molecular mechanism and then back to a new screen to identify a better drug candidate. So starting with a model of RAS-dependent cancer cell proliferation, researchers had earlier identified a drug that seemed to be effective in this model. So this study starts by trying to identify the molecular target of that drug. So they discovered that SCH51344 was acting through the gene MTH1, also called NUDT1. So this is a nucleotide pool sanitizing enzyme that helps prevent the incorporation of oxidized nucleotides during DNA damage repair and DNA replication. So you can see how this would be a great target for selectively killing cancer cells because they have a fast proliferation rate and a high level of intracellular reactive oxygen species. So these researchers then used codon-optimized gene synthesis from GenScript to express the NUDT1 protein for enzymatic assays and for crystallization studies, and they were able to get a really nice crystal structure. So then they looked for drugs that could inhibit MTH1, and they identified S-crizotinib as a highly specific low nanomolar MTH1 inhibitor. And this drug is particularly well poised for drug development because it was first discovered as a contaminant in a preparation of the R enantiomer of crizotinib, which is already FDA approved for clinical use, and the two compounds differ in only one chiral center. So we're going to keep our eye on this drug. And again, I recommend checking out this paper for its really elegant full circle approach, and of course the fact that the codon-optimized gene was used for different types of assays, including crystallization and enzyme activity. Okay, next, in April of this year, really before the current Ebola virus outbreak had started spreading, a group of researchers from the CDC published a study where they developed a high-throughput luciferase-based system to identify novel inhibitors of Marburg and Ebola viruses. So they ordered codon-optimized Ebola virus genes from us at GenScript, and they found that the codon-optimized support plasmids gave them a 2,000-fold increase in signal-to-noise ratios, as well as high sustained luciferase expression that was easily detectable within 24 hours after transfection for both their Ebola and Marburg virus systems. So they used those optimized plasmids for the remainder of their experiments, and they were able to identify a new potent inhibitor of both viruses. And I think their big hope is that their method of using many genomes and an infectious virus platform can be adapted by other groups for further antiviral drug screens. So really impressive improvement in sensitivity when you use codon optimization. Okay, next we'll move to humans. In this case study, this is from a really nice paper in Nature Biotechnology from last year, where they wanted to express a combinatorial library of talons for genome editing of any human gene. Now, talons, as you may know, are transcription activator-like effector nucleases, and the specificity of their targeting is determined by a central repeat domain that contains multiple tandem copies of a 34 amino acid long segment. And within that 34 amino acid stretch, only 2 amino acids vary. Those are the residues at positions 12 and 13, which are called the repeat variable diresidues. And those 2 residues are what determine the base specificity of the targeted sequence. So for example, the combination of asparagine isoleucine targets adenine bases, histidine aspartic acid targets cytosine bases, and the way you build up the DNA sequence you want to target is through these repeat variable diresidues in the tandem copies. So you can see, looking at the schematic here, that this central region contains a very highly repetitive sequence. So that it's a little bit difficult to work with. So these researchers asked GenScript to synthesize 84 different constructs for them, and Codon optimization was helpful here for solving a few different challenges. First, they wanted to limit sequence similarity between different constructs in their library. This was quite a challenge given how highly repetitive the sequence is, but we were able to limit the maximum sequence similarity between different plasmids to no more than 81%, which met their requirements. Second, they wanted to exclude rare codons in order to maximize translation efficiency in human cells, which we did. And third, they needed each of these constructs to be synthesized with 100% accuracy in order for the specificity of targeting to be intact. Now this is not as trivial as it may sound when you're dealing with highly repetitive sequences. However, in their pilot test of 124 constructs, they found that all 124 talons were active and disrupted their target genes at high frequencies. This is a really impressive success rate, and that reflects both the researchers' targeting strategy and also GenScript's expertise in accurately synthesizing highly repetitive gene sequences such as those that encode talons. So for a final case study, let's come back to E. coli, and we'll look at a case of metabolic engineering, which is a growing field within synthetic biology. So metabolic engineering uses a variety of techniques, including codon optimization, to turn bacterial or yeast cells into efficient factories for producing a desired protein or other biomolecule. So in this study, they focused on improving production of medium-chain methyl ketones, which are economically important molecules used in biofuel and flavor and fragrance industries. So instead of simply expressing one gene that encodes one protein of interest, here what they were doing was expressing a cassette of several genes to essentially re-engineer a beta oxidation pathway. They expressed a codon-optimized FAD gene in order to increase the influx of fatty acid substrates into their pathway. They also tinkered with acetate production pathways, which might be diverting substrate away from the desired methyl ketone products. They wanted to express an acetyl-CoA gene that was cloned from the gram-positive actinobacterium M. luteus, but that organism's genome has a very high GC content of 73%. So they codon-optimized that ACO gene for expression in E. coli, in part just to get that GC content a little lower. They made a lot of other adjustments, such as consolidating 2 expression plasmids into one and optimizing their induction conditions. And altogether, they were able to achieve a 160-fold tighter improvement in minimal medium. And that was made possible in part by codon optimization, which increased the expression of key enzymes within their metabolic pathway. So, now we've talked about what codon optimization is, and how GenScript's OptimumGene algorithm works, and we've seen it illustrated through case studies that used OptimumGene to solve different types of problems for diverse research applications. So let's move to the last part of the talk, which is how can you get codon-optimized genes? Well, there are a number of online tools that you can use to analyze a gene sequence to identify rare codons using codon frequency tables from different hosts. In fact, this tool on our website gets a lot of use, and it'll give you a report that shows the features of your gene, such as the distribution of rare codons and areas of high or low GC content. So you can manually make adjustments to your sequence and then use this tool to see how those adjustments impact the overall sequence. However, this obviously is focused on eliminating rare codons, which I hope I've convinced you is not the only and in many cases not the most important factor. So there's an easier and better way. You can request free codon optimization by sending your sequence to us, and instead of just analyzing the sequence you input, we will optimize your sequence for you using our multi-parameter OptimumGene algorithm. So to do that, we have a quick and easy online form which you can access on our website by clicking any of the buttons or links that say Get a Quote Now. So you can, on that quote form, you can input either a DNA sequence, ATCG, or a protein sequence using the one-letter abbreviations for amino acids. And then you'll be asked if you want codon optimization. If you select yes, you'll get this nice pop-up window that asks you to select the host expression organism, and it'll ask if you want to optimize over the whole sequence or just part of it, and it'll also ask if you want to avoid any restriction sites. If you have specific constraints or requests that aren't on this form, you can simply email us at gene@genscript.com or call us. So however you choose to get in touch with us, one of our PhD-level technical account managers will review your request and email you an optimized sequence within 1 business day. And you'll receive an optimization report that shows you the new optimized sequence and how it compares to the original sequence across a number of variables. So in this example, you can see that Codon Adaptation index has been improved from 0.59 in the original sequence to 0.83 in the optimized sequence. But you can see in the distribution map that there are still some rare codons allowed, especially in the 5' end of the mRNA, and that's probably in the service of optimizing other features such as mRNA secondary structure. So this graphic on the right shows the location of any predicted stem loop structures in your optimized sequence, as well as the calculated free energy of different regions of the mRNA. This information isn't usually included in our default report because most customers aren't too worried about it, but you can request it if you'd like. And if you want to change some of the parameters, or if you want us to run the optimization again, we'll do it for you as many times as you like for free. And once you're happy with your optimized sequence, the next step is to order it. So we offer a number of different gene synthesis services depending on your project's budget and timeline. Most people choose our standard custom gene synthesis, which has a starting list price of 35 cents a base pair and 9 business day turnaround time, so you can get your Codon-Optimized gene in about 2 weeks. If you need your gene faster, our rush service can get it to you in as few as 4 business days. If you're on a tight budget, our Gen Plus next generation platform can make genes starting at 23 cents a base pair. This platform is really intended for high throughput synthesis, so orders of at least 25 genes. But after we launched it, we got a lot of requests from customers to offer that low 23 cent a base pair price even for smaller orders. So we now offer an economy service. It takes a little longer because we consolidate a bunch of small orders to run them on a single chip together. But that's also available for you. If you need a really long gene, or if you need an entire genetic circuit for metabolic engineering or other synthetic biology applications, we're just now upgrading our GenBrick service to have a better price and turnaround time for DNA sequences up to 15 kb. We can do longer than that. In fact, we've worked with the Synthetic Yeast Genome Project to supply 170 kb of a synthetic chromosome for them. So if you have a need for larger genes, just get in touch and talk to us and request a quote. So we have these standard packages with service names, but the point I want to make is that really every project we work on for every customer is uniquely tailored to your needs. And we're not just a gene synthesis company. We offer end-to-end solutions to help you at any stage of the protein expression workflow where you might need help. So starting with deciding on your amino acid sequence. In many cases, you simply want to overexpress a wild-type full-length protein, so you can get the amino acid sequence from a database like ProteBank and have us optimize it and synthesize a single gene for you. No problem. But sometimes you might want to try different variants in order to find the one that works best. For example, you might want to express a truncated version because that can improve soluble expression for obtaining a crystal structure. Or you might want to compare a library of variants in order to find the one that works best. You might want to generate a mutant protein based on a known gain-of-function or loss-of-function mutation, or you might want a small rationally designed library of variants, or you might want a very large mutant library for protein engineering. So whatever your strategy is, it's pretty easy and cost-effective to get dozens or thousands of genes synthesized using our GenPlus high-throughput gene synthesis platform. And if you need larger mutant libraries for directed evolution, we offer gene variant library services as well. For the next step, choosing your expression system, our protein department offers a pretty unique service called Potential that will evaluate protein expression levels in small batches across several different systems in order to identify the expression host that's going to be best for you. So that's a really neat system that can save you a lot of time and money. And then in terms of designing your expression clone, today we talked about Codon Optimized gene synthesis. If you have other needs for custom targeting vectors, our molecular biology experts can work with you on that. If you decide that you're just having too much trouble expressing the protein in your own lab and you want to outsource the expression and purification to us, our Protein Services Department offers a number of different platforms using bacterial, yeast, mammalian, or insect baculovirus systems. We also have special services for expressing purified recombinant antibodies or fragments if you're interested in that. And then we also have some neat technologies for post-protein purification, so a refolding service, endotoxin removal services. And if you need characterization such as MALDI-TOF or HPLC or circular dichroism to determine purity and other characteristics of your protein, we offer specialized protein characterization services as well. So I now want to move away from describing our services and just point you to a few other resources that might help you learn more about how to optimize these different steps of the workflow. For creating mutant libraries for protein engineering, to help you with designing your amino acid sequence, there's an archived webinar from the summer on gene variant libraries with some tips on designing and constructing them, and again some case studies to give you some ideas maybe for your research. My colleague Leanne gave a great webinar earlier this summer on the pros and cons of different expression systems and different techniques for protein characterization. My colleague Kashev gave a great presentation that focused on protein expression in E. coli, and he talks a lot about the evidence for using different fusion partners or epitope tags to increase soluble expression, as well as discussing other sort of E. coli specific factors that you can optimize. And my colleague Leanne is going to be doing a webinar a couple weeks from now focusing on how to maximize the purity and yield of your protein. So I just want to come back to sort of wrap things up by saying I hope that I've piqued your interest in Codon-Optimized gene synthesis, but I want you to know that GenScript offers a wide range of custom services as well as catalog products that can also help your research. We're the most cited biology contract research organization. We create custom bioreagents, or we can perform custom assays to save your time and to fill in any gaps in your in-house technical expertise or capabilities in any of these areas. We know that a lot of our customers prefer to do their own DNA cloning and protein purification until the point where they run into problems, and then they outsource their difficult projects to us. So that's great. We love a challenge. Please get in touch if there's ever anything you think we might be able to help you with. So these are the references which you'll be able to access on the archived version. Now I just want to wrap up by saying thank you for attending. I hope you found this useful. Please complete the survey that will pop up on your screen when this webinar ends. If you have any questions or comments on what we've talked about today, I'd love to hear from you. You can email me at rachel.spear@genscript.com. This webinar will be archived on the GenScript website, and the other webinars that I mentioned are also available there for on-demand viewing. You can either watch the recorded webinar with audio, or you can simply download the slide deck. And whenever you're ready to request Codon optimization for your protein of interest, you can go to our website and use the online quote form that I showed you earlier, or you can simply email your request to us at gene@genscript.com. So thanks again, and I'm now going to check the chat window to see if anybody has submitted any questions that I can answer. And again, if you don't have questions now but you have them later, feel free to email me later. Oh wow, okay, we've got a lot of questions. Let me read through these real quick. How about the expression of viral genes, such as HIV-1 envelope in mammalian cells? Have you ever tested the codon optimization of those to check how your algorithm performs? So we may have optimized HIV-1 envelope proteins genes for customers, but we do not do research in-house on HIV-1 envelope genes. So we wouldn't have any in-house data that we could share, I don't believe. I'll definitely check. But if we have customers who are HIV researchers who have requested those, we're not really at liberty to tell you about the results because one thing that we're very dedicated to as a company is protecting the IP of our customers. So any project details are going to remain confidential. Okay, but I could definitely check their literature. You can probably do a PubMed search and see if any HIV papers have used codon optimization for that particular application. The next question here is, has GenScript ever studied the influence of the 5' UTR promoter-derived sequences on a panel of codon-optimized sequences for a given gene? So I will have to check with our R&D scientists about that. That's a really interesting question that I'm curious to know the answer to. That's the sort of thing that would probably be best addressed by using a combinatorial library approach, where you could create different different constructs that have different 5' UTRs attached to, the same or different codon optimized sequences to try to see where there might be synergies between the 5' UTR and a particular codon optimized sequence. But I don't have a quick answer for you on that, but I'll definitely follow up with you. Thanks for that question. Someone says, so how worried should I be of codon optimization messing with the proper folding of my protein if I've got a mammalian protein expressed in human cells? Or can this be optimized for as well, regardless of the protein? That's a great question. Ideally, you'll have some quick assay that you can do in your lab to check whether your heterologously expressed protein is actually properly folded to function the way that you need it to for your experiments, just to make sure. I think that one way we could address that concern would be to make sure that the distribution of rare codons in the optimized gene is not too different from the distribution of rare codons in the wild type gene, because that would sort of, to the extent that the presence of rare codons can slow translational elongation, can cause those the ribosomal pausing, that would be one way to address that. It may be that our codon optimization specialists who actually deal with these orders a lot have better solutions or additional solutions. But that's definitely it's definitely a great question. And I think it's something that most people don't worry about because they don't realize that it can cause a problem. But as with that Neurospora FRQ paper that I showed you, there's definitely evidence out there that it can be a factor that you should consider. So it's great that you're thinking about this. And I can say that the best thing to do would be to request codon optimization and express that concern. And our specialists who are used to filling these orders may be able to give you some insight on that. But to play it safe, you might want to order a few different codon optimized variants and just test them all side by side. And you definitely want to make sure that you have an assay to make sure that the protein you're getting expressed is functioning the way you want it to. Okay. what is the overall effect of sequence optimization during insertion into a host? So we generally think of the effect of sequence optimization in terms of the comparison between the optimized sequence and the non-optimized sequence. And, you know, compared to inserting a non-optimized sequence, your optimized sequence should be expressed more efficiently. If you're asking about the step of actually transfecting or transforming your cells to actually insert the sequence into the host, I wouldn't think that codon optimization would affect your transfection efficiencies or your transformation efficiencies depending on your system. That's probably going to be determined more by your protocol for inserting the plasmid into the host. So if you're doing, liposuction or some other method, I wouldn't think that codon optimization would change your transfection efficiency or your transformation efficiency. Okay, and then, two people ask, how can I download the slides, or will the PowerPoint be available after the webinar? Yes, it may take a few days before they're up on our website. If you have an urgent need to get these slides, please email me directly at rachel.spear@genscript.com, and I'll be happy to send them to you post-haste. Okay. After the 8 o'clock webinar this morning, we did this morning, and somebody already emailed me asking them for them. So I was delighted to learn that people find this to be a valuable resource. So that's all the questions that I see for now, and we're getting close to our target time. So I just want to say again, thank you for attending. Please complete the survey so that we know how helpful you found this. Please feel free to contact me with any questions, and I hope that you'll join us for our future webinar archive, or our future webinars, and I really hope that you'll reach out to us whenever you need codon-optimized genes for your experiments. Thanks, and best wishes for your research.